Hi everyone, I'm Leonora, and today I'll be talking about a machine learning method to try to improve the resolution of our images. So first of all, just a brief recap of quantitative susceptibility mapping, which measures the spatial distribution of the magnetic susceptibility from the signal phase. And we can actually use this difference in susceptibility to identify and segment our vessels, as well as estimate Venus oxygen saturation, which I think after Nick talk, I don't need to re-explain what it is. So we already know. And we also know that in order to do that, we really need a very good spatial resolution, because we can have, like, with very low resolution, we can have a lot of partial volume problems. Therefore, we cannot really then segment our vessels and estimate OVF. But at the same time, in order to have high resolution data, we know that we need to keep the people in the scanner for longer, and also really need high field system. So it would be cool if we can actually reconstruct the high resolution image from a low resolution image. In general, the super resolution methods in machine learning are those methods that try to increase the spatial resolution. Deep learning outside the MRI has actually achieved very good results in image reconstruction, denoising, super resolution segmentation. And just like a brief recap on how deep learning works. So we got these multiple layers, and what we give to the model is just the input and the output. So we don't define the hidden layers. The layers are related one to another through some linear transformation. And when the linear transformation are convolution, then we are going to talk about convolutional neural networks. We have also some non-linearity in the models, which are the activation functions, but I'm not going to go through that now. MRI, a few years back, there was this paper from Chen that they have used this model, which is called deeply densely connecting neural networks. It's the DCSRN model. It's a convolutional 3D model to increase the resolution of the images. They use the Human Connectome Project dataset, the T1 weighted images, which is like available for anyone. And what is important here is that it's, first of all, it's a 3D model. So we are not transforming the basic characteristic of the images, because we are working on a 3D image. Is a densely connected, which means that each layer is actually connected to the previous layers, and it's not the sum of the previous layers. So this means that the model is a bit lighter from the memory point of view, and also this means that the training is a bit faster. So we decided to see if we could actually apply this model to the QSM images. So what we want to do, we want to try to increase the resolution of the image, and hopefully the partial volume problem is going to reduce. Hopefully the segmentation of the vessels is going to improve as well, and also the quantification of the Venus oxygen saturation in the vessels. How do we do that? We take this model, the VCF model, and we apply that to the QSM images. We cannot just apply, sorry, I did say something wrong, but we need to train and fit the model again. So first of all, for people that work with machine learning, you know that one of the main problems is actually take a model from someone else and try and make it work on your machine. And so we actually decided to see if the model was working on the same, exactly the same data set. As I said, they have used the group of Chen have used the Human Connectome Project data set, which is available. So we downloaded the same data set, and we downloaded the model, and we spent really months to make it work on exactly the same data set. But after we got very similar results to what they published, we decided, okay, it's time that we can now move on our data set. And yeah, so it's just MP-range, and the voxel resolution was at 0.7 meters. So our data set, as Kevin said, we have this one data set available in Kubrick, which is a really nice data set with a lot of different imaging techniques in there. And we just took the 101 multiple phase images. They were acquired on the 70 system, and the resolution is 0.67 millimeter. From these images, we reconstructed the QSM map. So we went through the phase unpropping, projection onto dipole fields, and nonlinear dipole inversion. And once we got the QSM maps, we created our low resolution images, and we created them by cropping the K-space by a factor of 2 in each direction, which simulate what we would have done on the scanner if we were acquiring low resolution images. And also it's what they did in the paper. So we just decided to do what the Chen paper did for their data. We did some pre-processing, and again, we used some data documentation, and then apart from the data documentation, all the other steps was actually exactly the same of the paper. So we splitted our data set into 70% of data for the training, 15% for the validation, and 15% for the test set. We normalized our images, and then we created these 200 random patches of the size of 64 by 64 by 64. So now we have our low resolution images. We got the target, which is the high resolution. And what the model needs to learn is just to go from the low resolution to the high resolution. And what the model is producing is a super resolution image. And what we are interested in is the comparison within those two. So there are some numbers in here. As I said, the first part of the work was actually make the model working on the same dataset. So on T1 weighted images from the Human Connectome project dataset. There are some metrics, which are the structural similarity index, the peak signal to noise ratio, and normalized root mean square. Those metrics are all metrics to actually compare two images. And as you can see, the DCSRN model is actually doing a better job compared to the other common interpolation methods like neural networks by cubic, nearest neighbors by cubic and cubic. And also we achieved, after months again, better results than those published in the Chen paper, because they had the similarity index around 0.93. Then for the QSM maps, also the results were much better compared to other common techniques, although they are not good as the T1 weighted, but still like pretty good. And also they didn't share, like on the Chen paper, they didn't share the hyperparameters. So we need to do like some work also on optimizing the hyperparameters. And as you can see, they were the best parameters were very similar apart from the learning rate. Yeah, so focusing on the QSM results, you can see in yellow the low resolution image, so the input to the model. The in blue is the super resolution, so it's what the model has produced, and the values have been plotted in the range of 0.2, 0.6 ppm, which is what is conventionally accepted to reflect the vessels, basically susceptibility in the vessels. So if we plot one on top of the other, you can see that it's actually doing a kind of a nice job reconstructing the vessels. What is also nice, if we take the super resolution, so what comes from the model, on top of the high resolution, which is our gold standard, we can see that some of the noise that it's present in the high resolution is not present in the super resolution. So which means that the model is also doing like a kind of a nice job in improving the noise. Then we used the brain charter, which is a tool to segment the vessels. And yeah, you can see here we did this vessel segmentation separately for the low resolution and the super resolution. So you can see that the low resolution is in red, and then underneath we just decided to plot to the super resolution in green. And what is nice is that you can see that some of the vessels are actually, it looks like they have been reconstructed, especially now the video is gonna zoom in, and it's like in here. Now we're gonna wait for the video. And like in here, it's nice that it looks like that there is a missing part in the vessel that the model was actually able to reconstruct. Like in here, I don't know if it's visible, yeah. Okay, then we plot, so the histograms that you see here are the histograms of the susceptibility values in the three different images. We got the low resolution in purple, the super resolution in green, and the high resolution in yellow. And you can see that the distribution of the super resolution is actually more similar to the high resolution compared to the low resolution, which is good because it's our target. And we guess that the higher susceptibility values that we see around 0.1, they may be due to the partial volume problem. Also, we just converted the susceptibility maps to the OVF maps just to show that we were able to map them OVF was in like smaller vessels. And then what we did is to plot the OVF values against the vessel diameters. So again, the brain charter tool that we use for the segmentation is also giving us the vessel diameters. And you can see on the x-axis that there are like different vessel size that were plotted against the different oxygen extraction fraction values. One of the main points here is that you can see that the oxygen extraction fraction estimation is actually more accurate for bigger vessels. So again, it leads us to the problem of the partial volume. And also what is nice in here, if you focus on the first 2 points, especially in here, you can see that the low resolution, because due to the fact that it's a low resolution image, had like a worst estimation of the oxygen extraction fraction. And the estimation was better in the high resolution, and also the super resolution has the kind of a similar good estimation, which means that the bias in the estimation of the OVF was actually kind of reduced after we used the DC-SRM model. So altogether, we can say that the DC-SRM is a promised architecture to reconstruct QSM images, as we have produced like kind of a very good super resolution images. The cortical vessel segmentation has improved, as well as the oxygen extraction fraction estimation, and that the partial volume effect was a bit reduced. And yeah, thanks to the people that contributed to the work, especially to Stefano, that he's in the audience, and we share the authorship of this book. So, yeah. Thank you.